In this video, the Hershey and Chase's bacteriophage experiment will be discussed. In 1952, 8 years after Avery, McLeod, and McCarthy's transformation principle experiment concluded that DNA was the transforming substance, scientists Alfred Hershey and Martha Chase published a paper titled Independent Functions of Viral Protein and Nucleic Acid in Growth of Bacteriophage that concluded the key finding that DNA is the source of genetic material. You might be telling yourself, of course DNA is the source of genetic information. It's DNA. What else can it be? But scientists were wondering whether it was DNA or protein that was the source of genetic information. And the reason why we now know that DNA, not proteins, is the source is because of Hershey and Chase's bacteriophage experiment. The experiment itself was actually quite simple. There are two batches of E. coli, one with sulfur 35 and one with phosphorus 32. The two batches of E. coli were infected with T2 bacteriophages, which are viruses that attack bacteria, are composed of proteins and DNA on the inside. In the experiment, this allowed for all the offspring in the sulfur medium to have proteins labeled with the radioactive isotope, and all the offspring in the phosphorus medium to have DNA labeled with the radioactive isotope. After allowing the bacteriophages to infect the bacteria, Hershey and Chase sheared the protein coats off using a blender, and after centrifugation, they found out that radioactivity was not found in the phages reproduced in the sulfur medium, but in the fluid, which indicated that the protein had not been transmitted to the offspring phages. In the phosphorus batch, however, radioactivity was found in the phages reproduced, which indicated that DNA had been transmitted to the offspring phages. In conclusion, it was DNA, not protein, that entered the bacterial cell. To summarize, sulfur 35 is in the protein and phosphorus 32 is in the DNA. And the key finding of this experiment was that DNA is the genetic material, not protein. All thanks to Martha Chase and Alfred Hershey.